All right, AP Bio fans, here we go. Another sharp short. This one on linked genes. One of the things we've looked at is some genes appear to be inherited together. So here's an example. And what we're looking at, of course, is as we know, a chromosome can have hundreds or thousands of genes. Some will be closer together, some will be farther apart. Linked genes are those ones that are located close enough together to usually be inherited together, and that's because, as we'll see, the crossing over rarely happens between those two. The break in trade usually is rare in that because of their proximity on the genes together. Anyhow, here's our example. We have a parental generation that is all wild type. As you can see, the female is all normal, wild type, gray body, normal wings. The male is to use the term recessive, the double mutant, black body, vestigial mutant. And when you cross those into parental generation, you'll get a heterozygous uh, female that will, well, heterozygous in this case, will take a heterozygous female who, with the dihybrid cross, who displays a wild type and gray normal wing uh, phenotype. Now, as we said before, how do we know the heterozygous way back with Mendel and purple and white flowers? We do a test cross. And so, again, with a male double mutant, we do a test cross. Now, think about it. We talked about this in Mendelian genetics. We would expect, um, really, in a monohybrid heterozygous, homozygous cross, you would expect to see 50-50. So, you would expect to see half heterozygotes and half um, uh, double mutants. You know, and that's the same dihybrid cross or not. So in a dihybrid cross, then you would expect to see basically a fourth of every option because you should have a half of the gray bodies, half, you have a half gray body, half black body option. You have a half normal wing, half vestigial wing option. It should give you about a fourth of each. Well, as you look at the results down there, what you see is you have a vast majority of what are called the parental phenotype, and only a small number of are what are called the recombinant or non-parental phenotypes. Instead of seeing 25% of each, you see very dominated by the normal parental phenotypes, the heterozygous and the homozygous double mutant, and only a small that kind of mixes both. Okay? So instead of seeing a 1-to-1 ratio, there was independent assortment. But many more. Okay. So instead of having a 1-to-1 the 25% of each in independent assortment, many more look like the parents in the recombinant. But some do. So what does that mean? Morgan found that body color and wing size are usually inherited together in specific combinations. These genes don't seem to assort independently. And he reasoned that they were on the same chromosome being inherited together. However, since there was also non-parental phenotype, you had to explore the theory of genetic recombination, producing offspring with combinations of traits that are not the same as other parents. And if you combine the findings of Mendel and Morgan and chromosomal basis of inheritance, then you have the basis for recombination. Okay? Remember, Mendel observed the combinations of traits in some offspring different from either parent. So, terminology here: offspring with the phenotype matching one of the parents are called parental. Offspring are called recombinants. Okay? And you would always see a 50-50 frequency of recombination for any two genes that are on different chromosomes. You got a 50-50 shot. So what do we see is going on here? Well, we see our friend crossing over. So the body and the vestigial or non-vestigial, whatever, the body and the wings are close enough on the chromosome that our old friend crossing over shows us where you get the non-parental combination, but they're so close together that their odds of crossing over is small because you don't often see breaks in it. And so what you can calculate is a recombinant frequency between the two genes. Okay? So you take the sum total of the recombinants, and in this case 206 and 185. You take the sum total of the 2,300 total offspring and get that percentage. And what you see is a 17% recombinant frequency. So you didn't get the 1-to-1-to-1, 25% of each independent assortment. You had 83% parental phenotypes and 17% recombinant. So Morgan discovered that genes can be linked, but the link was incomplete because some recombinant phenotypes were observed. Because yes, you can still have crossing over. It's just going to be a much more rare occurrence the closer they are together. So proposed that some process must occasionally break and change the genes, and that was the crossing over of homologs. Okay. So recombinant chromosomes bring alleles together in new combinations in gametes. Random fertilization increases even further the number of variants that can be produced. And this is the raw material on which natural selection works. Now, Sturdivant was one of Morgan's students and constructed and could use that recombinant frequency to make a map and orderly list the loci of where each trait is. His prediction was the farther apart two genes are, the higher probability the crossover will occur. So the closer they were least, and so therefore the higher, the farther the distance, the higher the recombination frequency. Okay. So that's what a linkage map is. A linkage map is a genetic map of a chromosome based on recombinant frequencies. Distances between the genes can be expressed as map units, or as he called them because of his boss, centimorgans, and every 1% is recombinant frequency. So in our last example, the wings and the body at 17% were 17 centimorgans or 17 map units apart. Okay. Now, it's a relative thing. It's not an exact science. It's not a precise location. What you can do with is order them. But as we'll see, we can find the distance between a couple of genes. But if we have like 3 genes together, they're 2 separate recombinant frequencies. Say the one on the right hand to the middle and the one on the left hand to the middle are not necessarily going to add up. And you'll see that even in the body wing thing, while the total was 17%, if you throw another gene in there in between the two, you'll see that it's not quite, the two halves don't add up to 17%. That's why it's not a perfect science. It just helps you order things. Okay. So, oops, I'm going in the wrong direction. Hold on. All right, there we go. So the less, yep, I think I'm in my, okay. So the less frequently crossing over occurs between 2 genes, the closer they would be. So don't expect that difference to match. And there we see Cinnabar eyes in the middle. It's about 9% recombinant between the body and Cinnabar eyes. We are about 9.5% between Cinnabar and vestigial wings or wing shape, and the two add up to more than 70%. So it gives you the order. It doesn't give you exact distances. Genes that are far apart on the same chromosome can have a recombination frequency near 50%. So while they are on the same chromosome, they behave as if they were different. And so you have to be under 50% to be considered linked. Sturdivant used those recombination frequencies then to make a map of fruit fly genes. So cytogenetic maps indicate the positions of genes with respect to chromosomal features. So I'll let you look at this and do a little practice on it. we see that A and B are 8 math units are 8% apart, A to C 28, A to D 25, B to C 20, and B to D 33. And then your job is to pause right now and see if you can figure out the order. Thank you for pausing. If you did it correctly, then you would notice that, and I don't care whether you put D on the left or C on the right, but D, A, B, C or C, B, A, D, either way is obviously the 8 math units were the two that were closer together that put A and B together. Then you had to kind of look at the A versus C and the A versus D and the B versus C kind of thing and get that figured out. And so here's some map units or centimorgans to give you an idea of things in the fruit fly. Notice that if they're over 50%, they behave as if they are not linked, as if they're on two different chromosomes, because two different chromosomes have a 50-50 shot. All right. And how we do that is with 3-point crosses. Okay. What you see here is we have our alleles, one set of alleles, all capital, all so call them dominant, and the other set of alleles all recessive. Okay. You will get some with the parental gametes, and you can have crossing over between A and B, and that's called a single crossover. You can have crossing over between B and C, that's a single crossover. And you can have crossing over both between A and B and C. That's called a double crossover. And those are the events you use to determine the order of genes. Okay. So what is the order in gene distance and distance between these genes? Notice that the most prevalent numbers, the highest numbers will go to the parental phenotypes. The lowest number would be the double crossover. That's the least opportunity. Okay. And then single crossovers will occur between the end and the middle. How do I use this information of how many I observed? Well, determine the parent phenotype. That's the largest number. Then determine the double cross. That's the smallest number. Okay. And compare the parents and the double crossover, because once you compare the parents and the double crossover, that will help you determine what gene is in the middle, which letters in the middle. And that's how you get the order. Then determining the distance is just figuring out the percent recombination. So between two of those letters, like between A and B or B and C, you take all the single crossover numbers and don't forget to include the double crossover because it also crossed over there. Divide by the total number of progeny times 100 and draw the map. So in this case, what we saw in the double crossover was, if you look at the parental phenotype and you look at the double crossover event, you only notice that you notice a switch. That C had to be the one in the center. Sorry, here we go. I was on here. C had to be the one in the center because between the double crossovers, it was the only letter that switched place. So her parents had capital ABC and lowercase ABC, and my double crossovers went capital AB and lowercase c and lowercase ab with C. That had to be in the middle. And then I could just do the percentage for each. Okay. I have a couple other examples, but we'll be doing those in class, and you have practice work on Schoology. So once again, that's our excitement on how to figure out recombinant frequencies and do gene mapping with linked genes. Have a great day. We will talk to you later.